Olá a todos, boa noite, sejam bem-vindos a mais uma das nossas aulas no curso internacional de Neurociência Cognitiva, tá? Espero que todos estejam bem. Deixa eu olhar aqui rápido, eu não tive tempo de olhar o chat antes da aula, tá meio ocupado aqui, que eu já sinto o clima do pessoal aqui, mas de uma forma geral, aparentemente está tudo ok. Eu saúdo a todos aqui para a nossa, se eu não estou enganado, tá? Eu acho que é a 29ª aula, já perdi aqui a contagem. Então, grande prazer poder ter todos aqui. Vou aqui passar diretamente a convidar o professor José Aparecido para a gente poder apresentar o nosso convidado de hoje. Professor José Aparecido, boa noite. Temos mais um convidado internacional hoje. Nosso curso é internacional de fato, né?
Boa noite.
Boa noite.
Boa noite.
Boa noite.
Eu gostaria
de dar um destaque especial à presença do Dr. Michael Miller.
Mas antes
eu vou falar alguma palavra.
Ao longo dos meses
de agosto,
setembro e
outubro,
compartilhamos dias muito
especiais.
Todavia, hoje é um dia
muito mais especial para mim,
porque estamos recebendo
um brilhante,
inteligente
e criativo colega,
professor
Michael Miller, da Universidade da Califórnia,
de Santa Bárbara, um dos campos universitários mais belos que conheci ao longo da minha trajetória acadêmica. E olha que
eu conheço um.
Aproveitando o momento,
quero agradecer
meus amigos
pesquisadores,
Jack Loomis,
John Foley
e Walter Gogo, esse último já falecido, com os quais compartilhei, além de fortes laços afetivos, muitos estudos em percepção do espaço na Universidade da Califórnia, em Santa Bárbara. Saudosos
tempos.
Quando avanço nos anos
que ainda me restam,
mas retornando em alguns momentos num passado ainda vívido, me lembro afetiva e, sinceramente, com algumas lágrimas caindo de meus olhos, ainda não cansados, as minhas caminhadas no campus da UCSB, ao longo das praias, da biblioteca, do bookstore, da Harvard Cafeteria e dos amigos de então. Como eu disse, é um dia muito especial. Estamos recebendo o Dr. Michael Miller. Quem é o professor Michael Miller? Ele tem interesse na área de cognição, percepção e cognitive neuroscience. Ele é graduado em psicologia da San Francisco State University, em 1994, e começou o seu PhD, os seus estudos de doutorado em neuroscience na Universidade da Califórnia, em Davis, onde foi premiado como jovem cientista. Posteriormente, ele se juntou ao Dr. Michael Kazaniga, que eu já mencionei, juntamente com o Givago, que é o fundador, um dos rotuladores, o que criaram o termo de Cognitive Neuroscience. Então, o professor Michael Miller, hoje é membro do Departamento de Ciências do Cérebro e Comportamentais na Universidade da Califórnia, em Santa Bárbara, e dirige esse centro, que é o Sage Center for the Study of Mind, e editor do ano dedicado à neurociência cognitiva, que é uma revisão anual publicada na Academia de Nova York, Academia de Ciências de Nova York. O professor tem publicado inúmeros papers. Para aqueles que gostam de fatores de impacto, o fatores de impacto do professor Michael Miller é bastante elevado. Não é? Eu posso indicar a vocês, olharem no Google Citation, vocês verão que os papers, os trabalhos são bastante citados. Então, hoje, quero dar as boas-vindas, Michael. You are very welcome for sharing with us your talk. Please. Seja bem-vindo. We are very happy you are with us today. Thank you very much. Michael.
Thank you. I really appreciate that kind introduction. And it's a real honor for me to be able to speak with this group and to share some insights about split brain research, which I'll be focusing on today. I just want to make sure, can you hear me okay, Jivago?
Now I can hear you very well.
Okay, great. So, so what I thought I would do is kind of step through, and in the first part of the talk about some classic split brain research that's been done for the past 60 years. A lot of it by my graduate advisor, Mike Zaniga. And then toward the end of the talk, I'm going to switch gears a little bit and talk about some new directions in split brain research using more modern network neuroscience methods and what we can do, how those methods can sort of add to our understanding of the split brain patient and just a better understanding of the human condition, I think, in a broader sense. So to that point, I just want to kind of share some pictures of my collaborators. These are my recent collaborators in the Split Brain Project. This includes Lucas Vols from the University of Cologne, Christian Bien, who's the director of the Epilepsy Center in Bielefeld, Germany. This is where our most recent cohort of split brain patients are from and that we've been testing at. Here's Mike Zaniga down here. This is my student, Celine Becker, who's contributed a lot to this work. And my postdoctoral fellow, Tyler Santander, has also contributed quite a bit to this, the work that I'll be talking about. So callosotomy or split brain surgery. This is a treatment of last resort for epileptic patients. It's quite rare, but it's been around for about 80 years. And basically, this is a complete severing of the corpus callosum, which is the largest fiber tract in the human brain, which connects the right hemisphere to the left hemisphere. And it does it from about 250 billion myelinated axons. And it carries almost all the information from the neocortex of one hemisphere to the other. And what clinicians have discovered is that severing the corpus callosum can relieve epileptic patients of their seizures about 85% of the time. So it's like I said, it's a treatment of last resort, but it can be very effective for patients who don't have any other alternative to their treatment. Now, this was discovered back in the 1940s by surgeon von Wagen, who observed that he had some patients who had tumors across the corpus callosum and that seemed to alleviate their epileptic seizures, their drop seizures. So he performed his surgery in about 10 patients. Those patients were tested by neuropsychologists throughout the 1940s and into the 1950s. And the surprising thing is that basically they reported that there was nothing wrong with these patients, that there didn't seem to be any effect on their cognition, their motor control, other than some initial symptoms that went away after a couple of months. These patients seemed to be perfectly normal, and family and friends couldn't tell a difference. Now, this is in sharp contrast to the work that was being done by Roger Sperry, which was started a bit later, but Roger Sperry did a lot of work with animals and cats and found that severing the corpus callosum had some drastic effects on these animals. So there's a bit of a conundrum for a couple of decades about the difference between human patients and animal patients until Mike Zanika came around. Now, Mike was at the time, 1962, he was a graduate student of Roger Sperry's. And they had a surgeon there named Joseph Bogan, who was just starting to do these surgeries in Los Angeles. And Mike, for the first time, introduced some lateralized testing procedures, which I'll explain in a moment, that really revealed something quite startling, that these patients had very drastic disconnection effects, which I'll also explain in a moment. But that really changed the field, and it led to a whole better understanding of functional specializations between the hemispheres of the brain. And that research continues to this day. It also led to Nobel Prize awarded to Roger Sperry concerning the functional specialization of the cerebral hemisphere. So it really had a tremendous impact on the field. So I'm going to talk a little bit about that work, but then I'll turn my attention to, like I said, to the more recent cohort of patients that we're testing in Germany, and how we're using some network neuroscience using fMRI and other techniques to sort of better understand the synchrony that happens between the two hemispheres. So let me just start by talking a little bit about the testing procedures that are used to lateralize both the incoming information into the hemispheres to make sure that we isolate that information to one hemisphere only, and then the responses that we get, how we can lateralize the response that we get from each hemisphere as well. So in terms of motor control, and this is probably known to all of you, but the motor control is very well lateralized in the human brain. So the left hand is primarily controlled by the right hemisphere of the brain, while the right hand is primarily controlled by the left hemisphere. Now, this hemisphere control is more well established for the more distal muscles than proximal, but in terms of the hand movements and fingers, it's pretty much isolated to one hemisphere or the other. Same is true for vision. Vision is also very well lateralized. And in the split brain patients, this severing of the corpus callosum doesn't affect the visual pathways from the retina to the back of the head because they cross over at the optic chiasm, which remains intact. So all visual information travels to one hemisphere or the other. So if you're looking straight ahead, then all the visual information in the left side of that visual field was going to be traveling exclusively to the right hemisphere. And all the information in the right visual field travels exclusively to the left hemisphere. Now, this isn't broken down by the eye. So the information from the left visual field goes to both retina, but then it crosses over and goes strictly to the right hemisphere. So if you can isolate the visual information to one visual field or the other, then you can present it exclusively to one hemisphere and not the other. So it's sort of combining these two aspects of the brain. You can have some testing procedures with these patients in which you're isolating the stimuli and the responses to one hemisphere or the other. And this is just sort of a depiction of how this might work. So let's just say, for example, you have the patient looking straight ahead at this fixation point, and then you flash the word face to the right visual field. Now you have to flash this quickly, usually within 250 milliseconds, because you don't want the patient to saccade over to that space. Otherwise, it's going to both hemispheres. But if you do it quickly enough, then that information will only travel to the left hemisphere. Now, the left hemisphere can produce language. We know that language production in most of these patients is exclusively in the left hemisphere. So the patient can respond verbally and say that they saw a face. However, if you present that word to the left visual field so that it goes to the right hemisphere, you ask the patient what they saw, and they will say that they didn't see anything. However, we know that that information is still getting to the right hemisphere and being read by the right hemisphere, because if we put a pen and paper in that left hand and ask them to draw what they see, even though the left hemisphere said that they didn't see anything, that left hand, which is controlled by the right hemisphere, will actually draw a face. And I'll show you an example of that in this video coming up. Now, this is a video. What I'm going to show you is a video of a patient named Joe that Mike and I tested back in the 80s and 90s. And you'll see Mike Gazzaniga there being interviewed by Alan Alda. This is for a Scientific America Frontiers documentary film. I was a graduate student at the time doing the testing with Joe. And I think the video will kind of explain itself, but let me show that.
That's right. That's the idea. Okay, Joe, I want you to keep your hands out in front.
In an experiment that's now a classic in brain research, Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Ship.
Joe is being flashed a word only to one half of his brain. Words flashed to the right
Storm.
are seen only by his left brain, and Joe can report seeing those words just fine.
Piano. Good.
But when a word is flashed to his right brain,
Didn't see that. Okay. Joe, I'm going to ask you
but now watch what happens.
to draw that with your left hand.
You're gonna be lost.
Why don't you try drawing another picture of it over here if that will help you.
Oh, phone.
Oh, okay.
It's almost as though somebody has given him a secret communication.
That's
And
right.
now he knows that it's a telephone. Up until then, he was blind to it.
So that was about 30 years ago. So it was a much younger me doing the testing. But I have to tell you, it's a pretty extraordinary thing to be sitting with these patients and to see this happening. Basically, you've got two distinct minds within one head, and you're communicating and you're sending stimuli to that right hemisphere. But the left hemisphere, which is the one that you're communicating with, so when you're talking with one of these patients, you're speaking to their left hemisphere because that's the only one that can produce language. So the right hemisphere is basically mute, but it's doing its own thing. And obviously, the right hemisphere still has control. So it's still able to take in that information. It's still able to process it. And it's still able to control the left hand to the point that the left hand can actually draw a picture of what that right hemisphere saw. And while this is happening, it's like his left hemisphere, his talking left hemisphere, is like a stranger to this event. It's like somebody walking in and trying to figure out what it is that person's drawing. I mean, the left hemisphere is observing what his left hand is drawing and still trying to figure out what the heck it is he's drawing. And it takes him a moment to figure out that he's actually drawing a phone. And again, I don't think there's any kind of more powerful demonstration of how disconnected these two minds, these two forms of consciousness are in a single person. Now, this is what Mike Gazzaniga had discovered this way back in 1962, like 60 years ago. Again, when he was a graduate student of Roger Sperry's. And he had been, Mike he was an undergraduate at Dartmouth College and then went to Caltech to do to study under Roger Sperry. And he had been working on these lateralized sort of procedures. It occurred to him that maybe the missing piece in studying split brain patients up to that point, because again, the clinicians didn't think that there was anything different about them, is that you had to use these lateralized procedures in order for this to come out. And so Mike created these procedures and tested this with the splits that they had at the time, and indeed found that these two hemispheres were quite disconnected. And so he did simple kinds of tests in this original study. One's called a stereognosis test, where you just place, the subject or the patient close their eyes, and you place a common object in one hand or the other, and then ask them what it is they're holding. So they can only get the sense of the feel. They're not seeing it, but they can get a sense of the object by holding it. If they hold it in the right hand, that information is traveling to the left hemisphere, and it can say what it is they're holding. So let's say it's a comb. That right hand feels the comb, sends it to the left hemisphere. The left hemisphere has the ability to then say it's holding a comb. If you put that same comb in the left hand of the patient, that information is going to the right hemisphere, which is unable to speak. The left hemisphere, the speaking hemisphere, has no idea what that hand is holding and says, I don't know. But if you ask the patient to use that object in some way, that patient will take that comb and run it through their hair. So they know what the object is, they just can't say it. So again, it's two different minds operating very distinctly. Mike used to tell the story about how this patient at home, what it was like at home for him, and everything was pretty normal, but there were some peculiar things. So, for example, his right hand would pick up its newspaper, the right hand being controlled by the left hemisphere, but apparently the right hemisphere was not into reading the newspaper, and the left hand would take the newspaper and put it back down. And he would for minutes be trying to pick up the paper with his right, while his left hand would try to put it down. I had a patient that I tested years ago named Vicky, who talked about how after the first year of the surgery, would get up in the morning and get dressed, and she would walk to her closet. One hand would pick out one dress, and the right hand would pick out another dress, and she would stand there just stupefied, not knowing what to do, until her daughter would come along and sort of help her out. So again, you know, it was a really sort of stunning observation and report that this disconnection was so complete and so profound in these subjects. But there's something else that Mike had also discovered, and this was later on in the 70s, that this speaking left hemisphere that you are generally communicating with just has this tendency to want to rationalize behaviors that were actually being controlled by the right hemisphere that was not being controlled by the left hemisphere, but the mind just wants to have some kind of causal effect for things. The mind has a sense of control, of free will, and often will try to rationalize the behaviors of the right hemisphere. So, for example, we had a patient, Joe, actually. We could send the right hemisphere a command, like stand up, and Joe would stand up. He would follow that command. But when we asked Joe, why are you standing up? And again, we're talking to the left hemisphere. Joe wouldn't say, well, you told my right hemisphere to stand up, because he has no idea that command was flashed to the right hemisphere. But instead, his mind would quickly come up with an explanation for why he's standing. And you say, oh, I just felt like getting a Coke. So again, it's like he has to have, you know, our mind just sort of feels this need to have a complete narrative, a coherent whole to our conscious experience. And we often will fill in these sort of missing gaps with this sort of rationalization. Mike discovered this with Joe Ledoux back in the 1970s with this sort of classic split brain experiment. This was with patient PS at the time. And basically they would show him this picture. Again, he's looking at the fixation point. So the right hemisphere sees a snow scene. The left hemisphere sees a chicken claw. And then he asks the patient, PS, to point to the objects below that correspond with what they saw. Well, the left hemisphere, seeing the snow scene with the left hand, points to the snow shovel. The right hemisphere, seeing the snow scene, points to the snow shovel with the left hand. The left hemisphere, though, seeing the chicken claw, points to the chicken with the right hand. And then when they ask Joe, why are you pointing to those two things? He comes up with an immediate explanation. He says, well, I chose the chicken because of the chicken claw, and I chose the shovel because you need a shovel to clean out the chicken shed, right? With no mention of the snow seam, but it gives an explanation for why his left hand seems to be pointing at a snow shovel, which doesn't really correspond with the chicken, but he finds some, in his mind, he rationalizes some kind of explanation for why he would point to that. And this seems to be a really fundamental aspect of our conscious experience, that that we are very driven to like I said, to have a whole narrative, to have a coherent whole to our existence. And this is something that really comes out of the split brain studies. And, you know, I think it says a lot about sort of our conscious experience. This is something that Mike and I wrote several years ago in a chapter that, you know, consciousness is this local phenomenon in the brain that uses whatever information is available to unify the mind into a coherent whole. And we really seem to be driven to that. And again, it's something that really comes out with these split brain patients in a very kind of unique way. So that's a little bit of background on some of the classic split brain research. And of course, there are many, many other things that came out of that research, including a better understanding of some of the functional specializations of each hemisphere. The right hemisphere, as I mentioned, is very specialized for language production and for hypothesis formation and other kinds of cognitions. The right hemisphere also has some specializations, including spatial orientation and other things that it seem to be predominantly a right hemisphere function more so than the left. But in general, each hemisphere is able to do quite a bit, and there's a lot of redundancy between the two. But what I'd like to tell you about now is some more recent work that we've done, and I have to say that it's been quite a while since either Mike and I have been able to test split brain patients in the United States, because it's never been a large cohort. I mean, we never had more than 2 or 3 patients that we could test at one time. But the surgery, this callosotomy surgery, was stopped. They stopped using it in the United States in the early 1990s, and our patients died off. I think Joe died in about 2012. So we haven't had any other patients that we're able to test since that point. But we recently were put into touch with a group through some colleagues of mine in Germany, and who has started the surgery back up. And an important note is that the surgery actually is done in the United States, but it's done with pediatric patients, young patients who have lots of other developmental disorders. It's effective for treating their surgeries, but they're not very effective for as research subjects, because they have a lot of other issues that are going on. For us, the model patient is one who's an adult when the surgery happens and has relatively normal cognitive abilities, and both before and after the surgery. So this group in Germany started doing the surgery about 5 or 6 years ago and got in touch with us, and we began this research collaboration of testing these patients, which included putting them in the scanner and doing some fMRI scanning and analysis of their brain, which is a really sort of a unique opportunity to look at something that hadn't been looked at before in these split brain patients. So, like I said, these patients, were no longer available to us as fMRI was starting to rise in the field, and around 2010, 2015. Something happened in the field of cognitive neuroscience that really changed the nature of fMRI research, and that is the rise of network neuroscience. So prior to that, a lot of fMRI scanning and research was focused on trying to identify areas that were more active during a particular cognition, right? So you study looking at faces versus looking at other things, and you're interested in looking at the areas that seem to be specialized for face processing, but looking at the areas that are more active for looking at faces versus something else. But there was a shift in the field at the time, and that is that, like I said, around 2010, we started to look at the brain as a whole, and the brain as sort of a network of regions that are talking together. And I just saw a question pop up here. Could you comment on the effects of the disconnection in cases of surgery on patients with epilepsy? Right. So the effects of disconnection. So I'll back up and talk a little bit about that. So the effects of the disconnection are usually not real evident. There's some evidence of it right after surgery. So, for example, a patient may initially have trouble tying their shoe because that takes some fine motor coordination between the two hemispheres. They may have trouble buttoning their shirt. And doing some other things that require some coordination between the two. But they quickly relearn that. And usually that kind of symptom disappears after a few months. And it's probably done because the two hemispheres learn to coordinate that activity outside the head now. Now they coordinate that by looking at their shoelaces and coordinating it visually, instead of doing that within the head itself. And so a lot of the initial symptoms that you see due to the disconnection effect go away within the first year, unless you do these sort of lateralized testing procedures. Okay, so just to go back to the network neuroscience, just to kind of, I'm not sure how familiar you are with these with this type of fMRI work, but basically you can put an awake adult into a scanner and run a functional scan on that subject when they're doing nothing at all. So we call this just a resting state functional scan. So they're just lying there in the scanner, maybe looking at a crosshair for about 10 or 15 minutes with no instructions, no goals, or anything like that. But you're looking at the brain activity across the whole brain during that time. Now, every little voxel of space in that brain, you're getting a measurement of that brain activity in that voxel. And that activity is going to sort of fluctuate spontaneously over that 10 minutes. And what they discovered is that spontaneous activity that's occurring, that different regions of the brain, that spontaneous activity can be highly correlated across that 10 minutes. And these would be areas of the brain that are quite far apart from each other. And so what you can do is you can go in and you can seed an area, a certain region of the brain, and look at all the other areas that seed is correlated with across the brain. And what you find is you find these sort of different networks of brain regions that are color-coded here that all respond spontaneously, seem to be connected, functionally connected, and they're synchronized over this time. And it's been discovered that these different functional networks respond to different cognition. So there's a network that's devoted to vision, one to somatomotory, one to cognitive control, another to default mode network. So there are about 19 different networks that have been identified using this method. This just shows the spontaneous activity of that seed region with another region of the brain. Over here is a matrix of all the regions. So you can look at like 200 different regions across the brain and how they are all correlated with each other. And this is showing the, well, let me get to, Well, I'll explain the matrix in a moment in a little bit more detail. The other important thing I think to understand about this functional connectivity and how these different areas seem to be talking to each other, even though the subject isn't doing anything of any particular importance. But so even at rest, these areas are talking to each other. That functional connectivity closely corresponds with the structural connectivity. So there seems to be some constraint about how different regions talk to each other that depend on the white matter structural connections between those regions. And they don't necessarily have to be direct connections, but there needs to be some kind of white matter pathway between the two regions in order for that to seem to occur. And so this is known as sort of a structure-function relationship. This just sort of depicts, again, that these sort of unimodal areas of the brain, there's a really, really tight correspondence between functional connectivity and structural connectivity, and a little less so out in transmodal areas. But nevertheless, they have this organization. And in particular, the corpus callosum, which again is that big, thick bundle of white matter, also seems to have a functional architecture to it, in which it appears to be different areas of the corpus callosum seem to be connected through homotopic connections to these different functional networks. And this is going to become important in the new research that I'll talk about in a second. So what we did was we were really interested to look at how these networks are affected by by severing the corpus callosum, by basically cutting off the communication between the left and the right. And the reason for that is that a lot of these connections, if I back up for a moment, cross the two hemispheres. So this functional network, you see, is connected across the two hemispheres. So if you take a region here in the somatomotor cortex in the left hemisphere, it seems to be functionally connected to its homotopic region in the right hemisphere. So there's a lot of interhemispheric connectivity that's happening in the brain in a natural state. So the question is, what happens when you sever that connection to the brain? And we wanted to study that in this very unique population. So in order to look at that, we needed some controls for our analysis. We got the control subjects from the Human Connectome Project, which is a huge database of brain imaging data. It has thousands and thousands of subjects to it. We were able to take a large sample of these subjects, 100 subjects, run them through our analysis program to look at how the normal brain looks like in a resting state condition. And then we can compare that with our split brain patients. So just to orient you to what I'll be showing you, this is a matrix of the two hemispheres. And the matrix shows the correlations between the 20 different regions that are identified in each hemisphere. In this quadrant, it's left hemisphere correlated with itself. So this is looking at interhemispheric connectivity. The diagonal is the one area correlated with itself, which is meaningless. But right next to it is the other areas it's correlated with. This here is the visual regions, the different regions of the visual cortex that are correlated with each other, which shows a really strong connectivity with each other. And then as you go out are the different regions, like somatomotor, all the way down to prefrontal cortex. And as you can see, the regions that are close together have a really tight functional connectivity. The other quadrant, this quadrant here, which is the one that we're really interested in, is the correlation between one hemisphere and the other. So this looks at the correlation between regions in the left hemisphere with its homotopic region in the right hemisphere. And I think what you can just see by looking at this as a whole is that the connectivity with the regions in the opposite hemisphere are almost just as strong as the correlations within the regions within the hemisphere. So the interhemispheric connectivity is not much different than the intrahemispheric connectivity. And this is really important. So this shows the strength of the synchronization between the two hemispheres, how tightly coordinated they are in their activity. And this is just another analysis that looks at sort of what's called community detection. Just puts all the data in there and looks at how the data seems to organize itself into these communities that are color-coded. Again, this just shows that the communities sort of extends across the hemisphere. So you can see, again, there's a somatomotor network that organizes itself in the left hemisphere as well as the right hemisphere. And they're almost like mirror images of each other, the two hemispheres. So our hypothesis going into this is that for these full callosotomy patients, that the interhemispheric functional connectivity, modularity, synchrony, all these measures, which are key components of the functional architecture of the human brain, is going to be severely disrupted by having that corpus callosum be completely severed. Because again, the assumption is that functional connectivity is being driven by that structural connectivity of that white matter going from one hemisphere to the other. One of the more valuable things with this group of patients that we tested, though, and I'm going to show you data from about 6 different patients, is that we have a couple of patients that actually only partial callosotomy patients. These are patients in which the surgeon stopped short of cutting the corpus callosum, fully cutting corpus callosum. And the reason that this happened is because when the surgeon is making his cut, he's pulling that right hemisphere away to go in there to be able to make that cut of the corpus callosum. But that can sometimes lead to congestion in some of the arteries. And when that happens, that's a very dangerous thing. The surgeon will actually stop the surgery at that point. And so we actually had 2 patients in which that's the case, in which only a part of their corpus callosum was severed, which makes for another very interesting hypothesis, right? And as I mentioned before, the architecture, the functional architecture of the corpus callosum seems to be distributed from anterior to posterior based on these sort of functional networks. And so some of these functional networks are being disconnected while others are being left intact, at least the communication between the two hemispheres. So the hypothesis in this case is that for the partial callosotomy patients, that the disruption should be only partial, should only affect those regions of the corpus callosum that are affecting that sort of functional network. And then if you have a test to test that behavior that's affected by that, that you may see a disconnection effect, but that you won't see it in the areas that are still fully connected. And I'll show you the outcome. Now, that's a straightforward hypothesis, but what we found was actually quite surprising. So again, this just illustrates what I was talking about, that there's this anatomical gradient to the functional brain networks across the corpus callosum. This is the splenium, which is in the back of the head. So all the fibers that cross in the splenium are going to both visual cortices. So it's mainly visual information that's being transferred here. This is a dorsal attention network. This is a somatomotor network. This is a frontal parietal network and a default mode network. So as you go forward, you get more sort of executive control functions. As you're going behind, it's more basic visual, motor coordination, and the basic visual. So the surgeon always started from the anterior end of the corpus callosum and made his cut along here back to the anterior portion. So if he had to stop, what's going to be left intact are these posterior regions. And depending on where he stopped, as you'll see, we had one partial in which he stopped at about 1/3 of the way. And so he was leaving intact all the somatomotor pathways, visual pathways, and some attention pathways. But the other patient, he almost got all the way to the end. He got all the way to the splenium before he stopped the surgery. So in this one, you would expect that maybe some visual information would get through, and there wouldn't be a disconnection effect in terms of visual information, but any other behavioral disconnection effect should be evident in these patients. And so that was the hypothesis. And in order to test this behaviorally, so we did two different tests. One is we did a behavioral test to look at these disconnection effects across the spectrum of cognition. And then we did a test looking at the brain network itself in fMRI to see if the synchrony was indeed disrupted as would be predicted by these two different cases. So in terms of the behavioral testing, this is a battery of tests that my graduate student Celine devised. It looks at starts from the back of the brain, looks at the visual cortex, doing things like finger perimetry. Some of these things you can just do bedside. Some require computer testing. It all depends on the ability of the patient. Finger perimetry is just when you sit there, you have the patient look at your nose straight ahead, and then you move a certain number of fingers to the left visual field or right visual field and see if they can count the number of fingers, or if they notice that the hand is moving. So it's strictly visual. Then you have tactile, you have stereognosis, again, which is somatomotor. You can feel it in your hands, and that information then crosses over down to motor coordination, bimanual coordination, finger tapping, and so on. So there's a whole battery of tests that we can devise. Again, for the full splits, we expect them to be disconnected on all these tests. But for the partial, we expected them only to be disconnected on the ones in which that area was severed, that area of the corpus callosum was severed. So we expect in this case, since all the partials had visual pathways intact, that would be intact, but everything else would be disconnected. So I'll show you what we found. So this is a anatomical image of the 4 full callosotomy patients that we tested. As you can see, the corpus callosum in all these patients was completely severed and is completely missing. So it's completely dark there. And these are the 2 partials. And so this patient, as you can see, only that anterior third of the corpus callosum was severed, leaving this huge portion intact. And this was I call our star patient, who had almost the entire corpus callosum severed, except for this little bit of splenium. And it's about 1 centimeter of splenium. And what was interesting about this patient, when we went to Germany for the first time to test this patient, the clinical people at the hospital actually presented him as a full callosotomy patient, that they said, well, this is another full split. And so when we started to test him, we expected to see this disconnection effect. But we noticed actually within minutes of testing that this patient was not disconnected at all. He was actually seemed to be fully normal with no disconnection effects. And it was quite startling. And so we came back and tested this patient several times, as I'll show you, and found some really interesting things. So in terms of our behavioral testing and our hypotheses, so indeed, when we tested these full callosotomy patients, what we found was a full disconnection effect. As we predicted, they were disconnected on every measure that we tested them on, which is about 15 different measures, and they had the full disconnection. Now, as I said, what we expected, our hypothesis for the partial was a partial disconnection, that they would be affected on some tests, but not on others. But the surprise was this, that when we tested them on all these tests, what we found is absolutely no disconnection effect whatsoever. So even in our patient that only had a splenium of corpus callosum left, he showed absolutely no disconnection effect whatsoever. Just like a normal patient. It's like he had a full corpus callosum. All he had was 1 centimeter of the splenium left intact, and which really kind of dumbfounded us. Now, I should say that there is literature on partial patients from the past. So these patients have been studied for like 60 years. Some of them have reported partial disconnection effects. But the problem with those patients is they tended to be tested right after their surgery. So they're often tested within a few months of their surgery. And many of these patients then went back and got a second surgery in which the rest of their corpus callosum was fully disconnected. So really don't know what these patients would be like a year after their testing. Would they be able to recover these synchrony between the two hemispheres as these two patients appear to have? Because these patients were tested, this one was tested 6 years after his surgery, which may have been time for these networks to reorganize themselves. And actually that's what our current hypothesis is. So let me just show you the network data on these patients as well, because that's also quite interesting. So again, this is what you would expect to see in healthy controls. This is the connectivity within a hemisphere. This is the connectivity between the hemispheres. And this is just, of course, the mirror image of that. And I'll show you this compared to the full callosotomy patients. So again, this is the healthy controls over here. This is the community breakout that you would expect to see. And this is the first full callosotomy patient. And as you can see, their interhemispheric connectivity is completely disrupted as we expected. You do see a little bit of visual cortex connectivity that seems to be intact. It's a little less than the intra, but that's mainly because the patient has their eyes open during the scanning. So both hemispheres are actually taking in the same visual information, and there's some synchrony that's probably happening by the fact that they're taking in exactly the same visual stimulus. And we've tested this by having them close their eyes, and this will go away. But as you can see, for the rest of the brain, it's pretty much completely disrupted. All that synchrony that's happening in the normal brain and within their intact hemisphere. You can also see it in this community analysis. So again, you would expect to see these communities sort of disperse themselves equally across the two hemispheres. You do not see that in these full splits. This is not the mirror image of this as it is in a healthy subject. So it's a pretty distinct and robust effect. This is just this one full split, but you see this in the second one, same thing. You also see it in the third and in the fourth. So again, that interhemispheric synchrony is completely disrupted in these full callosotomy patients. So then the question is, what happens with the partials? As I said, behaviorally, they seem to be fully intact, but will we see that with this analysis? Will there actually be some disruption of the synchrony looking at their brain activity, as you might expect would happen with just a partial disconnection or severing of the corpus callosum? But what we find is that they, again, seem fully connected, that their synchrony does not seem to be disrupted whatsoever. So this is the first partial who has just the anterior third disconnected. And again, its connectivity doesn't seem any different than the healthy subject. So the interhemispheric connectivity seems completely intact when you compare it to the intrahemispheric connectivity of this subject. And what about the patient who has just a splenium, just a little bit of corpus callosum? What does it look like? Now, admittedly, it's a much more noisy data set with this patient, but again, the interhemispheric connectivity looks no different than the intrahemispheric connectivity. And even the community analysis, what you see in the left, you see the mirror of that image in the right. So even though he has everything gone in that corpus callosum except for that splenium, it appears that his functional connectivity and synchrony between the two hemispheres is completely intact. Now, our hypothesis going forward on this is that what we think is happening is that quickly within the first year, that these brain networks are learning to reorganize their connectivity, that the brain seems to be driven to have some synchrony between the two hemispheres, and that it may be reorganizing itself through polysynaptic connections and then crossing over in that splenium. The splenium turns out to be a really important area because it also connects sort of multimodal areas of the brain that are used to integrate information from different senses, and this could be a key region for having this happen. Now, whether we would see this, the surgeon went in the other direction, we don't know, but we hypothesize that this could be happening. And what we're doing now is we are now testing these patients prior to their surgery. So we'll do the scanning, behavioral testing preoperatively. Then we'll do another scan and testing session immediately after their surgery. And then we'll follow up every 6 months after that to see if we can track how these networks sort of reorganize themselves over time, which may give us some special insight into the nature of these patients. So in conclusion, let me just say that, and this is sort of our tagline for the splenium patient, is that it seems to be conscious unity hanging by a thread. You know, the thread of the splenium seems to be enough to sort of for this patient to have that sort of conscious unity between the two hemispheres, which we think is kind of extraordinary. But just as a whole, full callosotomy in the human brain produces two distinct minds. We've observed this for the last 60 years of testing these patients. The left hemisphere interpreter, that speaking left hemisphere, is continually trying to weave together sort of a unified conscious experience, right? Taking whatever information it has, and it often has missing information, but it puts together a story that makes our experience seem unified and whole. And we know that's a really important element of our conscious experience. But then lastly, that this synchrony between the two hemispheres, that it seems to be so important, and we seem to be driven to have that synchrony, can be achieved through relatively few callosal fibers, maybe after a period of reorganization. So thank you for your attention. I'm happy to answer any questions.
Hi, Michael. Hi, Michael. No, I think my microphone is not too good, but I will try to talk without echoes.
Yeah.
So fantastic lecture, Professor. Impressive condition of some people without corpus callosum. I will start with a question of mine to you. My first question is not about the patients with without corpus callosum, but you showed the synchrony between the hemispheres that the community people has, the healthy people has.
Yeah.
What you can talk us about the variability of this this connectivity between hemispheres? Is very big differences between us, or we are very the same, please?
Yeah, there's a lot of variability, and as you can imagine, there's a lot of variability between subjects and patients, but there's also a lot of variability depending on the area, and so there's actually been a lot of work looking at this in the healthy individuals to better understand what that variability might mean and what it contributes to. And so there's been some work looking at how that variability and connectivity is related to, let's say, the age of the subject, and as the subject gets older, or other kinds of issues. There's also work looking at, whether, cognitive ability and other kinds of things can contribute to some of the variability that you observe in the functional connectivity. And you can see even with the patients that I showed that there's quite a bit of variability from one patient to the next, which we always had to be careful about and take into consideration. But you know, this is something that while this is another area of research of mine, is trying to understand individual variability in brain activity across the brain. It used to be the case many years ago that variability across individuals was just thought to be merely noise, right? And there is a lot of signal to noise issues in fMRI research that has to be dealt with. But you know, when I started doing fMRI research back in the 1990s, it was all about trying to collect a whole group of subjects and collapse across that group so you can get rid of that variability. But that's actually, you know, that's actually not the case. That a good part of the variability between people is actually systematic, that there's something about the individual that leads to that variability that could be very important and needs to be studied. So I don't know if that answered your question, Divago, but I think that's a really important one. But
independent of the variability, the functional consequences are very low or nothing. Yeah.
Oh, you mean in terms of the matrices that you're seeing? Yeah.
Yes, the matrix. Yes.
That's
yes.
right. That's right. So there are some areas in which the visual cortex, for example, is going to have very little to no functional connectivity with, right? And that's generally how these sort of functional networks are identified, looked at, it just says something about the functional architecture of the human brain by looking at that. And there are all different ways to kind of analyze. I mean, this was the most simple kind of analysis that I showed you, but there are all different ways to kind of analyze that over time. But yeah, that variability could be really important.
No, voltei. Let's see. Yes. Now my internet was bad now. I have a question about from one of the people from audience, Amarillo, I think is Amarillo Carvalho. The question basically is it possible to monitor the interruption of this connectivity since the childhood?
Since the child,
how we, yeah. In the cases of congenital agenesis of the corpus callosum or something like this.
Right. So theoretically, yeah, I think it would, and actually that would be very interesting, is to look at, because with acallosal patients, these are, you know, children born without a corpus callosum. It appears mainly through anatomical studies, but looking at structurally, at that the rewiring seems to happen through different parts of the brain. So it happens sort of subcallosally. So a person that's born without a corpus callosum is not going to have a disconnection effect. They'll seem perfectly normal. But it would be, and I don't think this has been done, but it would be fascinating to look at the functional connectivity of these patients over time, right? To be able to look at, and it is possible to put, you know, an infant, for example, in a scanner and look at their functional connectivity in a resting state and measure that, and then see how that might progress over time. So that would be great.
How far this kind of knowledge is from the clinical application? I don't know in the US. In Brazil, it's very far. We don't have expectation to have something like this, but in the US at least.
You mean in terms of doing this kind of functional fMRI work?
Yes.
Yeah, so it's progressed pretty far. I mean, we have, you know, at our university, for example, we have a top of the line MR scanner in the basement of our psychology building. So so we do a lot of this functional connectivity work right in the basement of our, you know, right next to our laboratories. And You don't find this at every university here in the U.S., but there are several that have this kind of ability to not only do this with human subjects, but also to do this longitudinally with children as well as others. It's really, yeah, it's really key to this field to have that kind of availability because it can be quite complicated to try to do this in the hospital where you're competing with clinical scans and everything else.
Oh, good, good. I think Professor José Aparecido is without connection now. I will search for. I will try more question about you for you.
Okay.
The people here was today astonished with the lecture. The people had very few questions today, but
right.
What this kind of understanding about the connectivity in the interhemisphere is related to the people to be left-handed, right-handed.
Yeah.
It makes a difference for your kind
Sure.
of study.
Yes, it does, and you know but you know normally, and we haven't had a lot of these split brain patients that actually are left-handed, and even more rare are the patients that are left-handed but have language in the right hemisphere. I mean, typically when so it would be really fascinating to do the same kind of analysis on one of those patients. I don't have one of these. All the patients I'm testing are all right-handed and have language in the left hemisphere. What we would expect to see is basically the same thing, just everything kind of flipped, right? So we really wouldn't, in terms of the connectivity, we really wouldn't expect to see anything different with one of those patients, but it would be fascinating to try it out.
Yeah. Professor José Aparecido, if you would like to come to make a question, please.
Sure.
Feel free.
Yeah. Givago, I have a problem with my connection. Could you take from the private chat?
Yeah.
I think I made 3 questions, but the 2 first questions, Michael gave the answer during his presentation. So
Of course.
I think many people are asking from the general community, how about the future of cognitive neuroscience from his point of view, because he studied with Michael Casanegra, and Michael is founder of this area around the world. Many thanks, congratulations.
Thank you. So the future of cognitive neuroscience. Wow, that's a big question. But I think but I think we still have, there's still a big gap between our understanding of the brain and the cognition from outside of the head looking in to the cellular level, right? To how we know that neurons respond and act, and trying to make those 2 connections has been really difficult. And I think it's going to take, trying to close that gap, I think is going to be really important in the future. And again, I think it's just going to take an evolution of the techniques. I have a postdoc in my lab who is actually working with epileptic patients, but in a very different way. He's looking at patients that are in a hospital. They're preparing for surgery, and and before they have surgery, they have intracranial electrodes implanted in their brain to try to record seizure activity, right? And oftentimes these patients are there for a week or two. And I'm doing this in collaboration with some colleagues down at Cedars-Sinai in Los Angeles. And and my postdoc is going down there. And what you have the ability to do is use really sort of micro electrodes that are attached to these electrodes, and then be able to record from single neurons the activity in response to memory or decisions, and look at the different response types. So if we could be able to do that with these patients and somehow make that connection between these sort of grand pictures of functional cognitivity that we see in fMRI, and to see how that might be related to those single cell recordings, I think could really advance our understanding of brain function. But we're still a long ways from that. But that's the dream. That's the aspiration. I'd say that's the aspiration of the brain. In terms of, I'm also a cognitive psychologist, so to me the big question is consciousness. How does consciousness work, and how can we better understand that? And how can we operationalize it and look at it? So I think we're still far away from that as well. Javago, I think you might be muted.
Sorry. During the presentation, you showed some results about perceptual things. The people look at an object with one side and with the other no. But how about emotions?
Yeah.
What the implication of the loss of corpus callosum in emotion expressions?
Yeah, that's a great question. So emotions, the emotions is very much a subcortical process, and the transfer of that information travels subcallosally. So it may go through the hippocampal commissure or some other route ways. So typically, in our patients, if you evoke, let's say you show a stimulus that only the left hemisphere sees that evokes a certain emotional response, that emotional response will be felt by the right hemisphere. But the right hemisphere will have no idea why it feels that emotion. So you can end up with these sort of interesting situations. And Mike Zanika talks about a story in which he was testing a patient. He was going to the hospital to test the patient, and he was showing some really disturbing images to the right hemisphere of the brain. And it kind of evoked some negative emotions in the patient. But the left hemisphere didn't see any of this, right? But the left hemisphere felt those emotions and felt this sort of negative emotions, and actually but didn't know how to express it because they didn't know what's causing it, but they assumed that it must be Mike, right? So the patient actually was feeling really negative towards Mike because of these emotions that they were experiencing. I think, again, that interpreter just kind of rationalized why they were feeling those negative emotions and attributed it to something that Mike was doing. I think you're muted there. Again. Are you muted?
Sorry again. Professor José Aparecido, we are coming to the end. Do you have some point? I have one more question.
Sure.
Okay. George asked a very interesting question. He said, just for curiosity, was found the opposite excess of fibers?
I'm sorry, what's the question?
It was found before or during your experiment an excessive number of fibers in opposite of lack of fibers in the corpus callosum. Is there this possibility to find more fibers to connect both hemispheres than you expect, something like this?
Yeah, so it's a great question. I don't know the answer to that yet, but we are looking at it. We do have, I'll just throw this out there. We have a patient that we just started doing some pre and post-operative testing on. We did the first pre-operative testing last February. He had a surgery in February. We did a testing 2 weeks after, and then 6 months later. I didn't show his data because we're still not quite sure what's happening here. But in this patient, he did have the full callosotomy. We think the full callosotomy completely severed, but there appears to be some initial recovery of his hemispheric connectivity. And there seems to be some recovery of behavior that we're not quite sure about. And there seems to be some recovery of his seizures as well. So is there something that's happening? Are there some fibers happening outside of the corpus callosum that there's possible reorganization might be happening? We don't know at this point, but we think it's an intriguing case that we're going to be following closely. So I may have a better answer to that in the future.
So thank you very much, Professor Michael Miller. Thank you very much. Our course of cognitive neuroscience is complete with this kind of lecture, with the hometown, the place where the cognitive neuroscience comes. And we thank you very much for your participation in your course.
It was a pleasure. It was a pleasure. Thank you for inviting me.
Professor José Aparecido.
Yep.
Thank you, Michael, for
Thank you, Jose.
your excellent, magnificent talk. Please.
Great. Well, next time you're in Santa Barbara, let me know.
Some moment, give them my best, Ricardo.
I will.
I am missing them and their family. I hope I can see you in Santa Barbara someday.
That would be great. I would like that, and I'll let them know that I talked with you. Bye-bye.
Maravilha. Bye, Michael. Professor Jean, que aula, hein?
Oba! Isso,
o pessoal hoje ficou meio assustado, mas o assunto é pesado, eu admito.
Sim,
mas eu tenho certeza que olhando a aula depois... É incrível, incrível essa aula. Olhando a aula depois, com a legenda...
O Michael é da terceira geração. A primeira geração foi o Roger Spray, que ganhou o Prêmio Nobel de Physiology. Depois o Michael Casanega, que fundou lá em Santa Bárbara e nomeou a área de Cognitive Neuroscience. E agora o Michael Miller, não é, que é o novo diretor e pró-reitor lá em Santa Bárbara, dando continuidade a esses trabalhos maravilhosos.
Maravilha.
Parabéns a todos que têm nos acompanhado. Teremos novidades aí em algum momento sobre o impact factor, né? Vamos comunicar a todos.
Vamos comunicar a todos em breve. Tá? O professor está num congresso agora em Santa Catarina, no congresso da IBNEC. Então, aproveite a viagem. A gente vai ter agora um intervalo até a nossa próxima aula, devido aos congressos que vai estar ocorrendo agora em outubro. Mas logo, logo a gente está de volta. Um abraço a todo mundo e boa noite, pessoal.
Boa noite a todos. Abração, Givago. Obrigado novamente.